I'm super excited to welcome Dr. Josh Rubin today, and you can see his title of his talk right there. And before I start, I will give a land acknowledgement. I want to recognize that we live, work, play, and participate on a community, and that's not all of us here, but many of us here on this call on the unceded, ceded, and traditional territories of the 203 First Nations, along with the 38 Métis chartered communities, each of which possess their own unique traditions and history on the land that we now refer to as British Columbia. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration of the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of the work, we are committed to ensuring Indigenous women's rights to health and safety and an equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I'm actually not joining you from Vancouver, but I'll pretend that I'm in Vancouver right now, which I am headed there this week. So I guess it's not completely a lie, which is part of the unceded homelands of the Coast Salish peoples, the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. And with that, I'll just very briefly tell you about the Women's Health Cluster. We have over 400 members worldwide across 18 different countries. We run a number of events, such as the seminar series that you're joining us right now. We also have a hormonal contraceptives conference running currently, so please take a look at that if you're interested. And join us. It's free to join. Feel free to donate as well if you want to. It's not free to donate, but please feel free to donate if you want to, if you do. And with that, I'm going to stop sharing our screen. The last thing I should say is there are some trainee travel awards, so please do reach out and apply for those because we have very good success rates, unlike NIH and CIHR, the equivalent here. While I introduce Dr. Josh Rubin, he can start sharing his slides. Dr. Rubin is a professor of pediatrics and neuroscience at Washington University School of Medicine in St. Louis. He's originally from I should have practiced this before, Ardsley, New York.
Ardsley, yeah.
And is a graduate of Yale University. You might have heard of that. Dr. Rubin went on to receive his MD PhD from the Albert Einstein College of Medicine in New York City. And in 2003, he was recruited to Washington University, where he established his research lab and co-founded the Pediatric Neuro-oncology program at St. Louis Children's Hospital. In this clinic, his research activities include many therapeutic clinical trials and a significant focus on understanding the basis for cognitive deficits after treatment of pediatric brain tumors, a really important work. The focus of his laboratory is on links between normal brain development and the genesis of brain tumors, especially those aspects of development that render males more vulnerable to brain tumors than females, and the development of novel therapeutics. And I've heard Josh speak only once, but it was really fantastic. So I know we're all in for a treat. And his fun fact, as many of you know, I know intimidating listening to all of these accolades about our speakers. So it's nice to hear that they've had another life. And I love this fun fact from Dr. Rubin. He took 5 years off so those of you that are taking some time off between their undergrad and grad or medical school, you'll appreciate this. Between his college and starting his MD PhD, and during that time he performed Irish traditional music in Boston in a group called Finn McCool, and I'm really hoping there's a YouTube video somewhere.
There won't be, because it was 1982 to 1987, and Finn McCool is a mythological character, kind of like Paul Bunyan in Irish folklore. Seemed like a good name for a band.
It's an awesome name for a band.
Well, thank you, Lisa, so much for the invitation. I'm incredibly honored and excited to be here today. And thank you for your introduction. And today we're going to be talking about the special challenges that exist in translating the spectrum of sex differences in cancer biology into sex-adapted treatments. And I have nothing to disclose. So we're going to, hold on. That's funny. There's a slide in between that slide and this slide. Hold on 1 second. I must have accidentally deleted it. It's okay. It was just to tell you what we were gonna cover. And I can tell you, we're gonna start off with just very briefly talking about sex and gender so I can establish the way that I'm going to use those terms in this talk. Then we're going to talk about sex differences in cancer, incidence, outcome, response to treatment. We're going to talk about why it shouldn't be surprising that there are sex differences in cancer based on the evolution of sex differences and the developmental mechanisms involved in their genesis. And then we're going to talk about the challenge. And I'll leave that where it is for right now. So sex and gender. Under normal circumstances, sex refers to a biological phenotype that arises through sexual differentiation, initiated by being either XX or XY. Throughout early development, and actually throughout life, there are chromosomal effects that persist. These most dichotomously involve the non-pseudo-autosomal region of the Y, which is just a very few genes, but some of them are highly cancer relevant. And then those genes that escape X inactivation, and that's about 15% of the X chromosome in females and humans. About 15% that include a network of genes that interact with the p53 pathway, the master tumor suppressor, a network of genes that are part of innate immunity, and several key genes that are part of epigenetic regulation. So throughout life, the p53 pathway, innate immunity, and epigenetics are different from the time of fertilization on in male cells and in female cells. Parts of the difference include sex-specific reprogramming of imprinted loci. So you can say that sex differences really might start earlier than fertilization. And then, of course, during in utero development, there is the epigenetic effects of in utero gonadal hormones. And as testimony to how critical that is to sexual differentiation, individuals who are XY make normal testes and make normal amounts of testosterone, but have a mutation in the testosterone receptor, have what's known as androgen insensitivity syndrome. And those individuals are born looking phenotypically like a normal female. They have intra-abdominal testes that can cause them problems, and they don't go through puberty. But the magnitude of the in utero epigenetic effects of testosterone are really just remarkably illustrated by that condition. And then, of course, there's the activational effects of circulating sex hormones, which begin at puberty. And as a consequence, there is actually a spectrum of sex-specific phenotypes, from maximal XY effects and maximal testosterone effects to maximal X effects and maximum estrogen effects. And a significant portion of the population exists in between those maximums. And this concept of there being a spectrum with a male pole and a female pole is going to be very important throughout the course of the talk. Gender, in contrast, refers to where an individual places themselves in between two culturally defined poles of male and female. And obviously gender can also be not just where an individual places themselves, but also whether or not they place themselves on that spectrum between culturally defined poles of male and female. Sex and gender can be completely aligned, as in cis, or opposed, as in trans, or anywhere in between. Non-binary genders have always existed in human populations. Anybody who tells you that this is a fabrication of liberal, Western, modern culture is completely misinformed. Non-binary genders are normal, and obviously genders exist in a spectrum between culturally defined male and female and everything in between, including non-binary. Okay, so sex differences in cancer. These are 26 of the top cancers that affect adults. There is no I have no intention that you would read these, but it's male incidence rates, female incidence rates, and the ratio of male to female incidence rates. It goes from nearly 5 at the top for cancers of the oropharynx and tonsils, and down to 0.01 for breast cancer. What's important is this. These are all the cancers that occur more frequently in males, and these are the cancers that occur more frequently in females. This sex difference in cancer incidence rates is true all over the world. And it's also true regardless of genetic ancestry. I am not showing pediatric cancers here, but the same is true in pediatric cancers. And so what you have is a sex difference in incidence that it occurs regardless of age, so pre-pubertal, post-pubertal, post-menopause, all around the world, so not greatly affected by exposures, and in all the genetic ancestries, so not a function of genetic ancestry. It's independent. It's intrinsic to sex differences in biology. Brain tumors, which is what we'll be talking most about, glioblastoma. Brain tumors in general are more common in males. Everything to the right of this center line is a higher incidence in males with the incidence rate ratios, and everything to the left is more common in females. Meningioma is not truly a parenchymal brain tumor. It's a mesenchymal tumor. Those are membranes that cover the brain. They're more common in females, and they're highly responsive to female sex hormones. And then tumors of the pituitary, which is really neuroendocrine tissue. It's also true in dogs. So to the right of this line is the male to female incidence ratio for gliomas in dogs. And it's true whether they're spayed, neutered, or not. The difference declines a little bit with neutering, but it doesn't go away. Again, suggesting it's not just about sex hormones. It's not just about being human. It's something basic to being male versus female. Females also survive longer than males after standard treatment for most cancers. Again, everything to the right of the dotted line is an elevated hazard risk for dying in males for these individual tumors. The only tumor that's to the left is Kaposi's sarcoma. Remember that breast is here as a male higher risk because males do much worse with breast cancer than women do. It's not that there's more male deaths from breast cancer. It's just that their hazard ratio, their risk for death is greater than that of females. Females with GBM have a survival advantage. On the left is a relatively recent study from NYU in children with high-grade gliomas. They all received the same treatment in the same medical center in relatively the same period of time. So this difference in outcome is purely a function of sex. This difference in outcome is bigger than any single agent effect in GBM. So the sex effect is bigger than individual treatment effects. In adults, you see something similar. This is from the Ohio Brain Tumor Study. It's a small number of patients, but even in very large numbers of patients, up to 18,000 patients, you see a statistically significant difference in outcome as a function of sex. So why is that? There's multiple reasons why that is. We study cell intrinsic biology. We study interactions between tumor cells and non-tumor cells, and we study response to treatment. So here's a relatively recent study of ours in which we used patient data. And in this panel, we're using patient MRI data to which we applied an analytical algorithm that Kristen Swanson, a collaborator on this study, developed that allows us to calculate something known as tumor growth velocity based on gadolinium enhancing images. And what you can see is that through the first, second, third cycle of treatment, which includes radiation and temozolomide chemotherapy, females show a steady decline in growth velocity, while males show an initial decline and then it levels off. And in fact, so this would suggest that females are responding better to standard therapy than males. And interestingly, if we divide males and females up to those that have high and low growth velocity, because there's a range, right? And they're overlapping. So, but if we divide male and female up into high and low growth velocity during treatment, we see that for males, it doesn't really matter. There's no statistical difference between their survival in whether or not they actually show any initial response to treatment. In females, there's quite a huge difference in survival for those that show an initial response, the green line, versus those that show a weaker initial response, the purple line. So we're very interested in why that might be so. And it should come as no surprise, and thinking about evolution and development helped shape the questions that we ask in order to study the basis for the difference in incidence and the basis for the difference in response to treatment. With regard to evolution, we can talk about individuals across species without having to refer to them as males and females. If we talk about the big gamete producers, these are ova, and the little gamete producers, these are sperm. And we think about something that's known as the per gamete or progeny resource sacrifice. So for females, or big gamete producers, they are born with all the eggs they'll ever have. One or a few are available for fertilization at any given time. They expend all the personal resources on the gestation of fetuses. They expend most, if not all, the personal resources on the nurturing of newborns. Their per gamete progeny resource sacrifice is enormous. And so they are said to be resource limited in their reproductive success. If they can't ensure that an entire litter in most of the vertebrate kingdom can survive, then there's no reason to ever get pregnant because everybody dies, the mother and the litter. And so they have evolved traits with regard to reproduction that allows them, one, to make mate selection choices, but also traits that balance resource needs in mother and progeny and for longevity. And sometimes this means balancing the needs for viability versus growth. And there's an enormously powerful illustration of this in three great famines that occurred during the 20th century, one in China, one in Africa, and the best studied one was in Holland during a winter during World War II, where the Nazis surrounded Holland and wouldn't let any food in through the course of a winter. During that winter, there were a number of babies born, a number of babies in utero that then were born. And what's been true over all the years since, so 60 years, 80 years, there have been hundreds of studies. And the babies born, the males were fewer than normal, but those born were normal in size. The females were normal in number, but they were small for gestational age. The interpretation is that the strategy in a female fetus was to preserve viability at the expense of growth, whereas in males, it was to sacrifice viability for the purpose of growth. If we look at the small gamete producers, by the same analysis, they expend virtually nothing in personal resources in a per gamete or progeny relationship. They make millions of sperm like every single day of their life. Not quite, but you get the point. They're physically capable of impregnating multiple females every day of their adult life. They expend no personal resources on gestation of fetuses and through most of the animal kingdom on the nurturing of newborns. They're said to be ova or mate limited. And so they have traits that allow them to successfully compete with other males and influence female mate choice. The purpose of being brightly colored in all of this, or having a huge peacock tail, is to influence female mate choice and to scare off other males. The important thing to recognize about things like coloration or a huge peacock tail or antlers on an antlered animal is that these things decrease lifespan because they increase predation. And so those males that survive are interpreted as the genetically most robust, and that's part of the female mate choice. So we're going to talk more about this because this influences the regulation of cell growth. It influences the response to DNA damage and other environmental stressors, including treatment stresses, with regard to the preservation of growth in males versus the preservation of viability in females. During development, as we said before, this starts with imprinting before fertilization. In utero, there's a surge of testosterone that masculinizes XY individuals, can also masculinize XX individuals, particularly in a litter, which has a single placenta. That in utero surge works through epigenetics primarily. There are sex differences in metabolism from the moment of fertilization that persist throughout life that impact on disease and wellness. And if you look at the gonadal hormones, you can see that they vary between males and females across life. And if you're looking for diseases that are responsive to sex hormones, what you expect to see is prevalence and severity paralleling the changes in circulating sex hormones. Okay, with that as background, here's the challenge. So we see these very distinct differences in cancer incidence and outcome. We see lots of cell intrinsic and tumor stroma sex differences in biology, but how do you target it? So here is a group of college-age students from, I think it's the late 1950s, lined up according to their height. And can you guys see my cursor? Somebody? Anybody?
Yes, sorry, I was
Okay.
trying to find my unmute.
Thank you. All right. So in white are the females, in black are the males. Period of time when college attendance was still more males than females. But you can see that at the extreme of normal height, the shortest normal height is all females, exclusively females. At the extreme of normal male height is exclusively males. Of normal human height is exclusively females. And then there's this big overlap in the middle. And in fact, these distributions, even with thousands and thousands of males and females, are held up just the same. The peaks, the means, are statistically significantly different from each other. But within a sex, there's a normal distribution that spans much of the range, such that most individuals actually have overlapping heights. This is the challenge, because sex differences are not dichotomous like gonad differences are. Sex differences lie on a spectrum, as we said early at the start. That being the case, you can't expect to be able to treat patients with sex-specific therapies. It's not like one therapy is going to treat all females better than it treats males. And similarly, it's not like one therapy will treat males better than it treats all females, because male and female phenotype overlap. And we're going to illustrate this before getting to what I think is an approach to solve it. So we're going to start getting at this by looking at the work of a former graduate student, Lauren Bristol, who was following up on that observation of ours about better response to radiation in female GBM patients compared to male GBM patients. So here Lauren is looking at our mouse model of GBM, which includes deletion of neurofibromin, dominant negative P53, and treatment with a growth factor like EGF. In all cases in Lauren's work, you'll see at least 5 data points. Those 5 data points represent 5 different independent litters of mice from which she isolated male and female astrocytes. And this is key to understanding the importance of this. Litter to litter variation in the magnitude of sex differences is the norm. It's a consequence of litter composition and the position of females and males because of the testosterone. So to better capture the heterogeneity in human sex differences, we use at least 5 litters for most of our experiments so that we can capture a range in the magnitude of sex differences. So on the left, you just see the dose response curves for female in red, males in blue, and you can see that the females are statistically significantly more sensitive to radiation than males. We looked at the basis for that. And so in panel B here is a Western blot for cleaved caspase 3 as a marker of apoptosis. We actually used multiple markers, but I'm just showing this to you here. And you can look down at panel D for a second, and you can see the quantification of that. And there's actually very little apoptosis being induced at all by radiation, and there's no difference between males and females in terms of the amount of apoptosis that's being induced. We looked at cellular senescence, which is a therapeutic response as its terminal exit from the cell cycle. And we did that by using senescence-associated beta-gal staining, which is the blue that you can see here. And here you can see that in both males and females, there's a very significant increase in senescence in response to radiation, but they're highly overlapping between males and females across the 5 litters. We looked at proliferation, and male cells really don't slow down in their proliferation as a consequence of radiation. And we've seen that multiple times in multiple experimental settings. And females do. And that difference in proliferation you can see carried forward in colony formation when we've taken irradiated cells and replated them and let them grow for long periods of time. You can see the males more readily form colonies after irradiation with these doses than the females do. So we're interested in studying senescence. It's a state of irreversible cell cycle arrest. It's a cell-intrinsic anti-cancer mechanism in that it prevents the proliferation of cells with lots of DNA damage and cells that have excessive activation of oncogenic pathways. A number of different stimuli can induce it. The cell intrinsic component, if you look to the right with all the different inducers up at the top, ultimately all go through P21, sometimes P16 or P15. And so we had previously looked at the sex differences and the activity of P16 and P21, which are cyclin-dependent kinase inhibitors. So they inhibit the cyclins and the CDKs, which hyperphosphorylate RB, a master regulator of the G1 to S transition in the cell cycle. When RB is hyperphosphorylated, it releases a family of transcription factors known as E2Fs, and the E2Fs go on to induce the transcription of genes that are required for S phase entry and movement into G2 beyond it. So we had looked a number of years ago now at the regulation of RB in our model cells. And we did that by starving them of serum for 48 hours so that they become synchronized when we add serum back with regard to their entry into the cell cycle. And so in the males, you can see that by 10 hours after adding back serum, they were already noticeably phosphorylating RB. By 12 hours, it's even more. It accompanies a degradation of RB, which is, excuse me, commonly known to occur. In females, you see very little induction of RB over that same time course. The males are poised to initiate proliferation afterwards, growth, whereas the females take more time. And we looked at all three of the most prominent cyclin-dependent kinase inhibitors in cancer, which are P16, P21, and P27. So first at the RNA level on the left, you can see that in this case, adding another DNA damaging agent, etoposide, there's no induction of P16. There is a significant induction of P21 in both males and females, but now the female induction is highly significantly different than the male induction. There's no change in P27. There's zero P16 expressed at the protein level. And when we looked at the protein level, P21 was again significantly induced, much more so in females than in males. And P27 really did not much. So we had a story that was building around this primary regulator of senescence that indicated that in response to DNA damage, female cells induce more P21 and they undergo more senescence. This was not a consequence of differences in DNA damage, and we can quantify the amount of DNA damage in response to etoposide or irradiation by looking at the prevalence or the abundance of gamma H2AX or gamma H2AX loci, here a Western blot for abundance. And it goes up with DNA damage as it's a marker of DNA double strand breaks. And you can see the amount of it is really the same in males and females. If we look at the ratio of gamma H2AX to actin in the etoposide versus the DMSO conditions, you can see there's just no difference in males and females. So once again, we're looking at a female-specific response to DNA damage. Now, this is a complicated slide that I want to take you through over the course of a couple of minutes because it serves the basis for illustrating the obstacle in translating this to the clinic. So here, with many, many data points, again, drawn from 5 different litters of mice. Lauren is looking at the induction, first of P16, otherwise known as the gene as CDKN2A, versus senescence, which we'd already demonstrated occurred in both males and females, but maybe with some difference in females. And then P21, whose gene is CDKN1A versus senescence. And then the ratio of mRNA for CDKN1A and its cyclin-dependent kinase target CDK2 and senescence. And what I want you to take away from this is that P16 or CDKN2A, there's no correlation with senescence and its expression. And really in all of our experiments, CDKN2A really doesn't factor in the regulation of these responses. CDKN1A or P21 on its own is correlated in both males and females, though the slopes are different. And when we look at the ratio of CDKN1A to its target, CDK2, you can see that it's correlated in both males and females, but now the slope looks even more different. If we look at the protein level, At 24 hours, which is when senescence is being induced, you can see that the female and male correlations are quite different. The male is no longer significant at 24 hours at the protein level. The female is highly significant with a steeper slope. And if we look at 5 days, when senescence has now been consolidated, you can see that while it's significantly significant in both males and females, it's very different in males and females. And in fact, when we do a two-way ANOVA and we look for the interaction between sex and P21 CDK2 relationship to senescence-associated beta-gal, we see that sex has a significant effect on the correlation. And so what's important here, and what's important as we move forward, is to recognize how overlapping values are for males and females. What's different is how P21 CDK2 relate to senescence, which is the therapeutic response. And this is the challenge. I think you can appreciate looking at these graphs that you could never approach analyzing this question with a t-test, right? There's no difference in the t-tests between these. T-test is the inappropriate test for looking at this. It's linear regression and two-way ANOVA that provide you with an insight into what the actual sex difference is. And the sex difference is really about the relationship between P21 CDK2 and senescence. And this, of course, helps in generating new questions and new hypotheses to figure out why that's so. I'll finish this illustrative case by just showing you that P21 knockdown abrogates the sex difference in senescence. And actually, this is an exception to what I said before. It's only 3 litters. So here we have male Cas9 with and without irradiation at 8 gray. You can see senescence being induced. Here you have male P21 knockdown, again, 0 and 8 gray. You can see that knocking down P21 has no effect on male senescence. Here's female Cas9 expression, 0, 8 gray. Here's female P21 knockdown. P21 knockdown makes the female response look just like the males. So those linear regressions were showing us that females were more sensitive to P21 CDK2 ratio than males were, right? The slope was steeper. So now we're in a position to be asking about why they're more sensitive, and that's ongoing work in the lab. But let's think about how to think about this difference in the correlation, rather than thinking that there is a sexual dimorphism, a dichotomous relationship between the sexes, like ovaries versus testes, rather than thinking that, and rather than trying to model that, and rather than using t-tests to analyze data, we're looking at sex differences that differ in magnitude for both males and females across a spectrum. And this kind of biology has previously been addressed, actually, at least 40 years ago, by 2 evolutionary biologists, Crespi and Summers. And they were talking about growth differences between males and females throughout the animal kingdom. And you can see that illustrated here. I like to point out that male orangutans are 4 times the size as female orangutans, normal adults. And if you just put that in human terms for a second, you know, that's like normal sized males and females that are 1.25 to 1.5 feet, or normal sized females and males that are approaching 20 feet tall. This is an enormous difference in body size. And so how does that come to be? And why is it true throughout the animal kingdom? It's not because males went out hunting. That's not the answer. If you hypothesize that there's a female growth optimum and a male growth optimum, and that these serve those evolutionary purposes of resource preservation, resource allocation on the female side, and growth at the expense of viability on the male side, you might think about there being male-biased actions that push males to taller heights and female-biased actions that push females to shorter heights. And you would also think that for the population that exists in between the poles with overlapping heights, that for males to move away from their pole is going to involve increased female-biased action and or decreased male-biased action. And the same would be true for females. It's either increased male-biased action and or decreased female-biased action. These biased actions are molecular pathways that contribute, genetics and molecular pathways that contribute to size. So what we're going to do, rather than size, is we're going to think about this with regard to the pathways that drive cancer growth, the hallmark pathways of cancer. And we are going to take whole transcriptomes from patients, which is the goal of molecular tumor boards, and we're going to use them to establish what we're going to call a transcriptomic sex index that will exist for any pathway that we're interested in: cell cycle regulation, metabolism, glutamine metabolism, glucose metabolism, apoptosis, immunity, inflammation. We're going to establish the same kind of spectrum that we had for height, and we're going to place individual cancer patients at a precise location between those poles. And together with the pathway analysis, I'm going to illustrate this all for you, that we do, we're going to be able to make predictions about how any male or any female might respond to therapy, even if they have the same TSI values, according to that male-biased, female-biased paradigm. Even if they have the same TSI value or the same body size, they may have gotten to that body size or TSI value through different mechanisms. So transcriptomic sex index is defined as the Bayesian posterior probability of predicting sex based on the whole transcriptome. So a posterior probability is like this. If I told you that I was thinking of an individual that was 7'2", and I asked you to tell me whether you think they're male or female, you'd have a very high likelihood of being right if you said male. Similarly, if I told you that I was thinking of a healthy individual that was 4'11", you'd have a very high probability of being right if you said they were female. But you'd have a tremendously hard time being right more than half the time if I told you they were 5'6". That's what posterior probability is. And I'm going to show you how we do this so that it makes sense rather than in words. And this is the computational work of Will Yang, who I've worked with for years. So here's head and neck squamous carcinoma with a male to female incidence rate ratio of 2.75 to 1. In red circles up here in the upper left are female cases. In fact, we define female cases as having a low BNN value, Bayesian nearest neighbor value, because we establish males down here as being 1. And so you can see we can take a UMAP of all the transcriptomes and cluster it by similarity between the transcriptomes. So the males are like all down here and most of the females are up here, though you can see males and females scattered in between, just as we were describing. We can take those poles and we can now transform that data into a ridge plot. So here, rather than head and neck squamous carcinoma, I'm showing you all cancer. This is close to 10,000 cases. And you can see the distribution of the male TSI values and the female TSI values. Looks just like height, doesn't it? Skewed to the right for males, skewed to the left for females. And if you look at the whole population, it looks like the whole population has a normal distribution. But in fact, the distribution of males and females is different underlying that. If we look at two cancers with oppositely biased incidence ratios, esophageal carcinoma occurs 4.2 times more commonly in males, and thyroid carcinoma, which occurs 2.5 times more frequently in females. You can see that for the esophageal carcinoma, the ridge plot is shifted towards the right for males compared to the ridge plot that is shifted to the left in females, and actually includes a whole other cohort that's not even in the males. And you can see that for all of them, it's skewed towards the right, as it should be for something that has such a high male to female incidence ratio. And similarly, oppositely oriented is the male distribution of TSI values with thyroid carcinoma, the female distribution, which is highly skewed to the left, and then the all patient distribution, which is also highly skewed to the left. And not surprisingly, what the median value is within a cancer is related to the incidence rate ratio. So those median values that are quite high are the male biased cancers with the biggest difference. Those with median TSI that are quite low are the female biased cancers, and so on. So internally, this all seems consistent with capturing some element of the male-female biology. If we look at the pathways across the 26 cancers that are associated with the male and female poles, so this is not what's associated in the middle. This is what's associated as at the poles, because that's where we can hopefully identify what the target for male biased and female biased actions in cancer biology is. And I want to point out a couple of things in this heat map on the left. We'll exclude sarcoma for the moment because it is unique in being oppositely polarized, if you will, compared to other cancers. At the top of this heat map, you'll see a high degree of female bias in pathway activation, and you'll notice that this is all immune and inflammatory response. There's a second female cluster down here that is also all inflammatory response. So inflammatory response in cancer and immunity in cancer, except for sarcoma, is a highly female biased set of actions. And this, of course, is actually consistent with what we already know about female immunity compared to male immunity. I'll be done in just a minute. I know we're running a little past time. And you'll notice for the male pole, it's cell cycle regulation, which again, we know is consistent with what is, basically male, it's growth. And if we look at the numbers of cancers in which there's a male and female bias, so in terms of inflammatory response, there are some male cancers with elements of inflammatory response, but the female bias ratio and having the inflammatory response be a pole defining pathway is 2.75. It's much more common in females, as is allograft rejection, an immunity pathway, interferon gamma, an immunity pathway, IL-2 STAT5, inflammatory pathway, and then KRAS, which is really a growth stimulatory pathway. And I'm not quite sure why this shows up so strongly, but it does. Then there's IL-6, JAK, STAT3, another inflammatory response pathway, complement inflammatory response pathway, and TNF-alpha, an immunity pathway. So you can see that cancer in females is strongly biased towards immunity and inflammation, whereas the male bias is E2F targets, that transcription factor that drives cell cycle progression, G2M, so the required checkpoint signaling for entering into mitosis, and then oxidative phosphorylation, because male metabolism, particularly oxidative phosphorylation, is at a much higher rate than females in general. And part of that is that mitochondria, which are all derived from mothers, are less efficient in the male cell setting than in the female cell setting. So the amount of oxidative phosphorylation that's going on is high. So to finish, there's tons more to that analysis, but in the interest of time, to finish, I want to illustrate the way that we're thinking we could utilize this prospectively to stratify patients for treatment. So if we think about that inflammatory immunity spectrum from the female pole of very high immune activity to the male pole of less immune activity, and we look at it in lung cancer. So what's known in several cancers is that immune checkpoint inhibition therapy is more effective in males than it is in females. One of the interpretations of that is that female immunity is already maximally activated. So putting in an immune activator has basically no effect in females, but it has a big effect in males. So if we were to hypothesize that the immune signature that we see in cancers having a female bias relates to response to immune checkpoint inhibition in the following way. High, strong immunity is resistance to immune checkpoint inhibition. Weak immunity, like at the male pole, is correlated with sensitivity to immune checkpoint inhibition. On the female side, those at the pole have the strongest immunity and the greatest resistance to immune checkpoint inhibition. But as you approach the mid-range of TSI values, there are females that would exhibit a response relative to the other females. It may still be less than what the males at the pole would exhibit, but they have a chance of displaying a response. So if you had a checkpoint inhibitor that was an early stage clinical trial, and you wanted to prospectively address the hypothesis that TSI values would identify independently males and females that are more likely than other members of their sex to respond to immune checkpoint inhibition, you could hypothesize that those males that had no inflammation immunity signature in their cancers would have the strongest response, and that those females that are furthest away from the female pole would have the strongest female response. So rather than treating everybody, you could test in a future trial, in a trial in which you treated everybody, you could test this hypothesis, and that's something that we're working to do. Because if it turns out to be true, you have a stratification tool that's based on whole transcriptomes that identifies a quantifiable sex difference that may relate to treatment responsiveness. Hoping you guys will ask questions about that. I'm gonna, what we were gonna cover was a picture of a horse with a big blanket on it. What we uncovered is a picture of a horse without a big blanket on it. And so I think what we've uncovered is that sex differences in cancer are relatable to evolutionary selected traits to optimize reproductive roles. And ordinarily, I talk a lot about the regulation of the cell cycle in this, and I talk a lot about metabolism in this, but in the interest of time and the goal for today, I didn't. Sex differences accrue throughout life from the time of fertilization, and sex-adapted treatment are possible, and they may improve outcomes. Here's the last few iterations of my lab, and our funders, NCI, Petal the Cause Foundation, Children's Discovery Institute, and a number of other family foundations. And I am going to stop there.
Well, thank you so much, Dr. Rubin, for that excellent talk. For those of you online, if you can give a little emoji, that's always very helpful to speakers after giving a talk, and I can't get you all up for some reason, so I can't even see if you're giving an emoji, but I hope you're giving the clapping emoji.
But you don't have to. It's okay.
Yeah. And do please put in, there's a question already in the chat, but please put in any questions that you might have. I have a couple, probably on the more naive side, but that's going to be fun. Dr. A. Addy, do you want to unmute yourself and ask your question, or do you want me to ask for you? Because I can do that if you'd like, but we're a small enough group that it's fine to unmute as well. But I also recognize that not everyone is in a position where they can unmute. Sometimes people are looking after children because it's different time zones in different places. It looks like you can ask. Okay, I will do that.
Okay, go ahead. I do, but somebody has
it?
to repeat it.
Yes, I'll repeat it. There's a family history of aggressive prostate cancers developing at a young age increase the likely incidence of any female-specific malignancies.
So that's a fantastic question, and I don't know of any data to speak directly to it. There are some data to suggest that testosterone can be growth promoting in female cancers, and obviously females always have testosterone receptors and are always responding to the low level of testosterone that is available to them. So I think there could be a likelihood. Certainly, if early life aggressive prostate cancer is associated with the polyglutamine repeats in the testosterone receptor, which shift the dose response to the left, you could imagine that that would also enhance the responsiveness in females, because it's a polymorphism in testosterone receptor, which in those cases where testosterone was driving a growth phenotype would also occur with them. The other thing to always keep in mind about testosterone is its immune suppressive effects. So testosterone affects multiple cell types in the immune system and basically suppresses, particularly innate immunity, compared to the absence of a big testosterone effect in females and the presence of a big estrogen effect in females. So both things you could imagine could increase the likelihood of even a female-specific malignancy if there was a left shift in the dose response curve to testosterone, as does occur in some aggressive prostate cancers. But I don't know any direct data.
I want to follow up on something you just said, but what I was thinking initially was when you're talking about the different litters and why you look at that so you can avoid litter effects essentially. I did notice that in one of your slides that there was some extremes, right? So it was in a female group, it was really high. So did you ever look back to see what were the litter characteristics?
Absolutely. So Lauren,
it's related to testosterone. Do you know that?
So Lauren, who was just phenomenal, she went back and looked at the litter composition of every litter that contributed to our studies. And it wasn't just 5, because sometimes we pool cells from 2 different litters. And so the magnitude of sex differences is a clear function of the sex ratio of pups, such that the biggest sex differences observed are when female pups are the majority and male pups are the minority. Anything in which males dominate, any litter with more male pups has smaller sex differences. And that is presumably a consequence of what's already been described as the masculinizing effects of testosterone in litters, because they're single placentas and there's enough, I guess, sharing of blood that they see that testosterone. The other thing that has been examined, not by us, but others, is that the location of a female pup between 2 male pups influences how much they are masculinized. So in the presence of a male predominant litter, the females are partially masculinized, and the magnitude of sex differences in that litter is smaller than if the females dominate in the litter. And so we have litters that represent a whole range of compositions. And, we really do feel like that's an element of modeling the differences in humans.
Mhm. So there's one, we're out of time, but there's one quick question, probably not from Pooja. So I thought we should.
Yeah, this is a great question.
Yeah. And I'm also wondering, does it matter if, the pregnancy was with a male? Or...
So here's what I'll answer. So for everybody who's not looking at the chat, Dr. Biswas is asking whether there's any study showing that females who've never had children have less risk of cancer or more longevity than females with children. It's a very, very complicated question. But what we and others have done related to this is to study fetal microchimeric cells.
Mhm.
So in any woman who's been, it's not clear when these cells pass, but any woman who has delivered a child will forever have cells derived from that child living in their tissues. Those cells, fetal microchimeric cells, are capable of multiplying, and they're capable of trafficking to sites of pathology. And in some cancers, their circulating cell abundance is associated with poor outcomes, and in others, it's associated with better outcomes. So this is a mechanism that people don't frequently think about, by which pregnancy and delivery can impact on health and disease. It's a phenomenon that really illustrates the difference in the challenges to female immune systems as compared to male immune systems. I mean, placentas are fetal derived, so they're haploidentical to a mother. They invade the endometrium very similarly to the way that a cancer invades tissues, but they're tolerated. And then they have fetal microchimeric cells that are present in their tissues throughout life. And if it's a woman who's had children with more than one father, the fetal microchimeric cells are not even identically haploidentical to the mother, and yet they're tolerated at the same time that they can maintain immune function for pathogens.
So complicated, but so interesting. I'm very well aware that we're 3 minutes over time, and people have places to go, and I don't want to take up any more of Dr. Rubin's time. But thank you so much. I have a whole bunch of other things that I hope I get to talk to you about.
Sure, anytime.
So thank you so much. Lots of.
Thank you so much for happening.
Yeah, please.
Having me, not happening, having me, and for running this series. It's wonderful.
Thank you. Thank you, Jane.
All right.
Bye, everyone.
Bye.